to programmatically access the large amount of data that is stored within WormBase. So I'm going to talk about, sorry, the blue is not showing up very well. We have SimpleMine, WormMine, and ParaSite BioMart we're going to talk about. And I'm going to talk about how to use these, what their particular advantages are, and which ones you might want to use for certain types of queries. So first, I'm just going to dive into SimpleMine. You can find SimpleMine under the Tools menu, over here under Data Mining and Batch Queries, along with the other tools that I'm going to talk about today. Gene set enrichment analysis, Raymond will talk about it in a little bit. Once you arrive at the SimpleMine page, it's really just a single page with a search box, a number of different options, and then a series of checkboxes. Now, the only intention of SimpleMine is to be able to take in a list of genes using a variety of identifiers. Of course, we accept WormBase identifiers and gene names, but we will also accept UniProt IDs, TreeFam IDs, and RefSeq IDs. And basically what this does is when you tell it what attributes of each gene you want, and it just gives you a table where each row represents one gene and every column is an attribute of the gene. So you can choose which attributes you want. All of them are selected by default. You can click on the Set All checkboxes. This toggles on and off all of the selected attributes so that you can easily turn them all off and just select a couple if that's all you want. So just go through this list, see which types of data that you want as columns in your table, turn them on and off. Then you can query your list once you've pasted in your list here. You can also query from an uploaded file or query all elegans genes. We also have two options here. If you have a small list and you just want to quickly look at the results, you can do HTML and just see a table in the web browser, or you can download those as a tabs limited TXT file. Okay, that's it. So that's SimpleMine, and it's quick and easy to use, and we encourage you to check it out. And let us know if you have any questions. Next, I'm going to talk about WormMine, which is a much larger, more powerful database for data warehouse tool for accessing much of the data that is within WormBase. It is fast and it's powerful. It can do a lot of things. One of the complaints is that it's a little difficult to learn how to use. I'm going to hope to try to remedy some of that today, but again, we also highly encourage you to reach out to WormBase, email help@wormbase.org, or any of us, you can talk to any of us tonight in the poster session with the booth to ask questions about how to best use this. So one thing that's important to note is that users can create personal profiles to save their lists, queries, and templates, and come back to them once they log back into the WormMine system. It's easy to share queries and download tables and query results. The WormMine data that we currently have loaded into WormMine pertains to basically the WormBase core species plus a few others. So this isn't going to have a large list of species. I'm going to talk about WormBase ParaSite BioMart. ParaSite, the ParaSite site and the BioMart associated with it has a large number of genomes, talking about I think almost 180 genomes, most of which are flatworms and nematodes. The data we have for these species includes the genes, both non-coding and non-coding genes. We have sequences for genes, transcripts, and proteins, alleles, phenotype annotations pertaining to allele and RNAi perturbations, expression patterns and expression clusters, and then we have all of these ontologies loaded, including the gene ontology, the phenotype, anatomy, life stage, and disease ontologies. And there's a lot of powerful querying you can do with those ontologies to help you find the information you're looking for. Just briefly, these are the species that we pretty much have a full complement of genes for in WormMine. There are other species represented in WormMine, but those are mostly like human genes and rat genes that have orthologs in C. elegans or some worm genome. But these are the worm genomes that we have, and those are the ones you can take advantage of. So what are some examples of queries that can be run? And I say simple queries here only to mean that these queries you can run as a one-off query. You can do many more complicated queries by chaining series of queries and query results together to create a much more complex set of results that you might be looking for. So what can you do? So you can return all of the genes and their genomic coordinates for a given species. You can return all the human orthologs for a gene or a set of genes. I just want to point out that this is using Panther calls only, so if you come to the WormBase website and you see orthology or parology calls made using other algorithms besides Panther, you won't see those in WormMine, just as a heads up. You can find, for example, all of the microRNA genes for a given species, or all the ribosomal RNA genes, any of the gene types. You can find all the phenotypes associated with a gene or a set of genes. You can return all genes of a particular type within a specific genomic interval. If you want to know all microRNAs within the left arm of chromosome 3, you can do that. You can return all proteins with a given protein motif. You can find all protein sequences for a gene or a list. And you can return all tissues or cells in which a gene is expressed, or return all proteins from a species within a given range of molecular weight. That just touches upon the variety of types of data that you can get out of WormMine for the species that I just listed before. Now, WormMine has lists, and lists. It has a powerful list feature, which basically just takes sets of objects. For the most part, we'll be talking about lists of genes. So you can upload lists of genes, alleles, anatomy terms, gene ontology terms. You can save these lists to your personal profile so that when you log back in later, you'll have them saved. And another important thing is that you can perform list operations like list comparisons or performing intersection or union operations. And you can do things like perform list subtraction. You can take list A minus list B, or also take the union of A and B and subtract the intersection of A and B. We can also use lists to constrain on them in our queries. When we say we want all phenotypes for a particular list of genes, for example, you would save the list of genes in WormMine, and then in the query builder you would say constrain to only show me the phenotypes for the genes in this list that I created. So we can return, for example, all the anatomical entities in which genes that have a sterile phenotype are expressed, and we can do this using lists. Return phenotypes of genes that are expressed in the intestine. Return sequences of proteins that are expressed in a list of anatomical locations, or return subcellular localizations of all genes encoding transporters, for example. So WormMine can be found also in the tools menu, a few slots down from the SimpleMine button. This is the WormMine homepage. I just want to show some of the basic elements. So here we have templates. So you can click on the tab at the top to go to our flat list of template queries. And you can also find our template queries down here along the bottom of the page. And these are actually separate tabs of different topics. So you can click on genomics and see these template queries that apply to genomics. You can click on proteins or expression and see a different set of template queries. So for example, we might want to look at this particular template query where we input a transcript type and a species, and a particular species, and then we get back the genes that fit that criteria. So the default is to choose C. elegans microRNAs, and we can choose the drop-down for species and choose a respective species, or choose a different gene type or transcript type. And then we click on Show Results, and we get a table. And this is what a basic WormMine interMine table looks like. From here, you can see the data listed. You can use different features in each column. You can close the column. You can sort it. You can hide it, and then collapse it, and then re-expand it later. You can filter. This is a little filter or funnel icon. And then you can look at a column summary. It tells you a brief summary of all the counts of all the different entities in that particular column. We just restricted our query to looking for microRNAs and C. elegans. So you can see that the filter icons are highlighted in red, indicating that the filters are active. Next, I just want to talk about lists. You can actually import lists of genes or proteins right from the home page, or you can click on the Lists tab, and you'll be brought to the Upload version section of the Lists tab, in which you can upload a list of objects in here, like alleles, anatomy terms, but most often you'll probably be looking at genes. And then you can then click on View and see the list that already exists. There's a set of pre-canned lists that have been created by us at WormBase that you might want to play around with or look at, test the list processing tools. And then let's see. So these are, for example, right here, there's a set of operations that you can perform on lists that you select with checkboxes. The checkboxes are a little hard to see here. But for example, we can perform a union operation, intersection, subtract, or asymmetric difference. And so, for example, I have two gene lists here that represent all genes with a lethal or subtype phenotype via alleles as of the WS257 release of WormBase. And then we have, similarly, genes with a cell cycle phenotype. Now we can take these two genes, you'd select both checkboxes, you'd click on union, and it would ask you to name your new list. And then you would name that union of cell cycle and lethal phenotype genes. Or you can create an intersection, you can subtract or perform asymmetric difference. And then you would save those lists to your own personal profile and come back to them later. Next, I'm going to do a brief overview of the Query Builder tool. This is where a lot of the power lies in WormMine and being able to query. It's a little overwhelming at first, but I would encourage you to check it out and play around and get a feel for it. If you notice any issues, please let us know. There are a few outstanding issues that we're working on with the core inline development team. But in the meantime, please forgive us for some of these oddities. But you can click on the Query Builder tab. You're brought to this page, which is the Query Builder homepage. You can select which type of data type you want to query for. We'll select the gene class. And then you're brought to the Query Builder overview and model browser. Now, this model browser contains all of the attributes of genes that are indexed or incorporated into the WormMine system. At the very top, each class that you might browse in the model browser shows data objects. These are basically just strings of text or numbers or true, false values. So you can turn and you can visualize these. And then we have all of the associated objects that have their own attributes in WormMine that are also connected to genes. And so you can drill down into these by clicking on the plus sign next to them and look at their respective attributes. Now, just zooming in here, I just want to point out that we, for starters, we have at the beginning of every object class that has its own attributes, we have a summary button and a constrain button. If you click on the summary, it opens a default set of attributes that you might want to query for. If you click on constrain, you can apply basic constraints to say, I want to make sure that I get everything that has some value in this field, or I'm looking for elegans as a species, so you can just type that into a filter box. Right here, and you'll see this for any particular attributes, you can click on show, and that will bring, that'll pop this particular attribute over into the query overview section. So typically that's what you want to do. If you want that value for a gene, for example, click on show, and later I'll show you how you can constrain by clicking on the constrain button, which allows you to specify a particular gene, let's say, or a list of genes that you want to look at in particular. And as I mentioned, there's a little plus sign here. It indicates you can expand that because that is its own object class in WormMine with its own attributes. So you could say, I want all worm genes from all C. elegans genes, and I want all of their alleles. Or I want all of the alleles and the phenotypes associated with them. So after I've selected several options by clicking on show and expanding and then clicking on other attributes, we can start building a query here. And what we see here is I've asked for the brief description and the gene IDs and names, and all alleles associated with those genes and all phenotypes associated with those alleles, and I'm also displaying the organism. And now there's no constraints here, so it's just going to return all genes in the database unless I apply a constraint. So here I'm going to click on constraint, apply here, I'll click show results, and then I get a table of all eggle genes and C. elegans that have, and the phenotypes associated with their alleles. Briefly, I want to go through WormBase ParaSite BioMart. Now, BioMart is similar to our old WormMart. If you remember working with the WormMart tool, it's all based on the BioMart technology. And this is very gene-based, so basically you would give it a set of filters, tell it which genes you're looking for, and then for each of those genes, what attributes you want to see back. So we and this will apply for genes in nematodes and flatworms. We can also find homology across these species and homology in humans, for example. It's also easy to share queries and download tables of results. It can accept a wide variety of gene identifiers. It's more forgiving than WormMine in that sense. We can take in GenBank and CDI gene, RefSeq, UniProt, PDB, and RNA Central and Wiki gene identifiers, in addition to WormBase gene identifiers. So some queries that can be performed in BioMart. You can return all the coding sequences of all genes with an ortholog in humans. Return the UniProt IDs for transmembrane protein coding genes in C. elegans. Return 3' UTR sequences of all genes with a given protein domain. Return 2,000 base pairs upstream of all genes in a list. Or return all ribosomal RNA genes from the species of the Rhabditoidae family, a superfamily. Return all paralogs within a species, or return protein domains for all genes localized to the Golgi apparatus. Now, ParaSite is right here on the data mining and batch queries that we've seen already under the WormBase Tools menu. This is what the basic homepage looks like. You can click on New. This should be the default aspect of the way that you see it. Again, here's the query filter section and the output attributes. These are the options for query filtering. So you can tell it, I only want to see genes from these species within this region of whatever chromosome, genes that have certain identifiers, genes that are annotated with certain ontology terms, genes that have or don't have homologs or orthologs or paralogs in other species, and genes that have particular protein domains. So we're filtering down all the genes that are in here down to just the genes that we want to look at. And then we can go, and this is an important thing I just want to point out. It's a powerful feature. As I said before, there's almost 180 genomes in WormBase ParaSite BioMart. If you click on species here, you can open it up and it allows you to click on a browse taxonomy view. And in this case, it's a really powerful tool where you can drill down between Nematoda and Platyhelminthes branches of the taxonomy to choose and select checkboxes that apply to whatever, say, superfamily that you might be interested in. And on the right, it'll show you all the species that apply. You can drill down further to get a more specialized list, or you can go further up the list and get all species. Now, output attributes. This is what the output attributes look like. There's two options, basically, to create a data table or retrieve sequences, say, in FASTA format. You can specify that for the output, you want species and genome information, you want the gene identifiers, you want external database references, you want to know gene ontology annotations, the domains that exist, or any orthologs or paralogs that exist. And then if you retrieve sequences, we have this nice cartoon. It allows you to select with the radio button to the left of all these types. You can look for flanking sequences, you can look for unspliced sequences, UTRs, coding sequences, peptide sequences, and so forth. If you click on flank, for example, then you can enter how many base pairs or how many nucleotides of upstream or downstream of that gene that you actually want to recover. And so this is what an output looks like. In this particular query, I queried for transmembrane proteins from C. elegans. And I put out, and I say, return, for all those transmembrane proteins in C. elegans, return all the gene ontology annotations, term names that are annotated to them. The last thing I want to point out is that a warning: if you use Excel or other spreadsheet programs, be very careful, because all of these gene names will automatically be converted to dates, and they will be irreversibly changed into dates, and it's really annoying. So make sure that when you import as text format, not as general, to avoid this problem. So with that, I'll thank you for your attention, and please email us with any questions that you have. Thanks.